very exciting Technical Tuesday that we have for you this month with a very special guest indeed. Miriam O'Sullivan is from Amgen, who's the recently promotion that I'm sure is long overdue, but recently promoted to the Director of Quality Assurance at Amgen. And she has very, very kindly joined us today for today's Technical Tuesday, and she will be speaking about single-use technology in drug manufacturing. So thank you very much, Miriam, and welcome today. For those of you that are familiar with the format, and for those of you who aren't, Miriam will talk for however long she'll talk for, maybe 30 minutes or maybe 45 minutes or however long you want to talk for. And then afterwards we'll have a little Q&A session. So you can if you have any questions, you can save them for the end, and I'm sure Miriam will be kind enough to answer them. So without further ado, Miriam, please take it away.
Well, good evening and good morning, and depending where you are. So I welcome the opportunity to speak to you. Kieran asked me and kindly let me pick my topics. So just to give you some background, I'm obviously, I have an Irish name and an Irish accent. So I started my career in Dublin. I worked in BMS and then Wyeth, which became Pfizer, and then went to Amgen. So I'm with Amgen almost 10 years now. So as Kieran says, well overdue promotion. So just to give you some background, I've worked in many different areas. I'm working now in a drug substance facility in Singapore, and the unique thing about our drug substance facility is that we use single-use systems predominantly. We were the first licensed single-use system manufacturer for commercial drug product or drug substance in the world. So it's sometimes it's very good to be first, and sometimes it's not very good to be first. So I'll give you some of the pros and cons of being first and for single-use systems as well. I'm happy to stop along the way. I know we don't generally do slides. I have some images. If you want me to share them with you afterwards, I can. But let me give you an introduction to the vision, right? So the vision initially was back in I suppose my early days in Amgen. They were talking always about this manufacturing of the future and what does the future look like, and getting ahead of the curve in terms of technology and what that can bring. And I think that's always been the kind of Amgen way, is to try and lead the way in biologics manufacturers. And so and I'm going to read a little bit. I won't read the whole thing, but just let me read. So in November 20th, 2014, Amgen announced it had opened this biomanufacturing facility in Singapore. The plant that uses single-use bioreactors, disposable plastic containers, and continuous purification processing, and has a flexible modular design. The plant will use less energy and water than traditional plants, will have the same annual output as a conventional facility. It's much smaller in size and saves money in terms of operating costs. So this plant will enable higher production, greater accessibility to medications to patients, and we're reinventing the means of manufacture, said Bob Bradway, who's the CEO, right? So this is the vision. This is what the plan was. And I have to say, in terms of delivery, we fairly much have delivered that vision in ASM. It is a very, very neat facility compared to a stainless plant. So I've been to many stainless facilities. When I walked into the main manufacturing floor in ASM, I was stunned that so much throughput can go through a very small room, and it is a very small room. The advantages I think are outlined in terms of the vision, but one of the things that we've seen in terms of delivery is we're getting a higher titer, which is kind of a help and a bonus in terms of delivery. So I think what our vision was this a small space, very high throughput, very flexible design, something that wasn't wasn't going to be too disruptive if you want to introduce a new product. Having a system that is disposable, that is, I suppose, if you think about it, a lot of our stainless plants have certain open processing elements. So we don't have open processing elements. We do have a lot of single-use connectors. We have a lot of aseptic connections, but generally the product train is end-to-end. It's a closed system, and this allows a great amount of flexibility on the manufacturing floor. So I suppose most traditional facilities have a lot of stainless. They're very difficult to change quickly. They rely heavily on cleaning and steaming, and that is one of the big constraints. So what we were looking at with building this facility, and it was really an experiment on Amgen's part, but a successful one, thankfully, was to have this flexible modular design that would support rapid NPIs, that could give us the flexibility that we needed in our manufacturing process, and also have a very small footprint. So in terms of, I don't know, have any of you who are Singaporean based been to Tuas? So Tuas is the end of the world in Singapore. Singapore is a small place, but Tuas is the equivalent of the middle of nowhere. So we're based out in Tuas. We've got a nice facility out there where we make biologics, and we also have a synthetic manufacturing plant. So in terms of capital projects, you heard the vision was in 2014, and very quickly, for a capital project from a greenfield site, we were up and running. So I suppose the challenges in terms of building and building on that vision were getting this higher, tighter process that I've mentioned, having these single-use systems that give you the flexibility, a modular design and construction, connected processing. So where we could, it was end-to-end, and we didn't have to break and make connections wherever we could avoid that. Real-time monitoring on the floor, real-time testing on the floor. So rather than sending samples to QC, we did a lot of testing actually on the manufacturing floor. That's a little bit controversial with some of the agencies, so that has provided us with some challenges. And then in terms of, this is kind of a blessing and a curse to some extent. We have a reduction in the variation in our raw materials. We're very heavily reliant on raw material supplies, and especially single-use system suppliers. So that's led us to a little bit of maybe single point of failure, but I'll go into that a little bit more in the cons when I get to the end. So what I would say, we have a high cell culture productivity in a small facility footprint. So compared to our Rhode Island site, which a lot of our tech transfers come from, the footprint is maybe about a fifth of what they have. It's a lot smaller. We highly utilize single-use equipment, so 95% of our equipment is single-use. It's a closed operation. Everything is pre-sterilized. We have aseptic connections, so we can truly call it a closed processing system. We have an integrated manufacturing layout. So the way our facility is laid out is we have a kind of a U-shaped facility with solution prep on one side and manufacturing suites on the other side of the U. And how we connect those two is we have this central corridor, and we actually connect physically. We pass our buffers and our medias and everything in through totes in that central corridor. We fill them from the solution prep side. We wheel them across the corridor, which is a small corridor. It's maybe 30 feet or 10 meters wide. And then we just connect them up on the other side. So it's a very neat, very small design, very connected all the way through. And I suppose when we want to change, the idea is that it's relatively easy. So in terms of our suite, we have a weigh and dispense, a solution prep, logistics, corridor, inoculum prep, the CMS, the central manufacturing suite, and then with purification and equipment prep, all in one very small footprint. So we are, our manufacturing suite is ISO 9, so it's a very deep facility. We have a minimum gowning. We don't need to wear coveralls, except when we're in the high areas, which are the inoculation and the final pure. So a little bit more about the next generation. So what I would say in terms of our single-use systems, the challenges that we're seeing are largely related to the single-use systems themselves. So in terms of we use very, very big single-use systems. So I think when people think of disposable technology, they tend to think of on a small scale. We're talking about 2K bags, so 2,000 litre bags. They're very, very big. They're very difficult to inspect. They're very difficult to manufacture, and they have a lot of things in them. So they come with a built-in T-sparger, or they come with a different connectors for probes and for monitoring of the equipment. So they're quite complicated. They're very, very specialized, and they don't always come in as you'd like them to come. So we have a lot of incoming inspection failures. Because these bags are so big, they come in double wrapped, triple wrapped. They're hard to inspect in the warehouse, so they're normally on the manufacturing floor before we find the issues. So we have a lot of pre-use inspection failures. We have a lot of deviations related to the single-use systems. So some of the connectors don't always work as we'd like them to. Some of the tubing may be on incorrectly, and so we're heavily reliant on our vendors getting it right first time. When we do get the systems up, and they're quite complicated to put up and to assemble, so we're very highly trained staff in the area where we are assembling these bags. The possibility of leaks is way higher than with a stainless steel system. So we do have, at one stage it seemed we'd a leak every day, but we have a lot of leaks. And when they're early in the process stream, they're not so impactful. But when you get towards your 2K bioreactor, where you've got a full batch that's worth a lot of money, they become very painful. So we do have, I suppose, a concern about loss of product. Potential microbial ingress is another issue if we do have a leak. And because we're saying they're closed operations, they're all pre-sterilized. If we have a hole in them, then that's very hard to stand over. So we have to be very diligent about how we investigate leaks. And the aseptic connectors, again, they're great, but if they don't work or they're leaking, then they're not aseptic anymore. So this is one of the challenges. In terms of other issues, we do have single source suppliers. So we have a lot of we designed the bags with the suppliers. We were the first in the market, so they're very customized. They're very specialized. We have a supply issue, a potential supply issue for something like COVID, where we can't get access to our single-use systems. We stop. There's nothing we can do. We can't improvise. We can't go a different way. We don't have backup stainless equipment. So we are, to some extent, at the mercy of our supply chain in a way that maybe other biologics plants are not. We are also very reliant on the vendors to be aware and cognizant and working with us to address any issues. So we find, there's a particular bag that's very difficult to put up. It's a 2K bag. It's very, you can imagine a 2,000 litre plastic bag, and you're trying to put it up without making sure there's no damage, there's low leaks, and there's a lot going on there. We found one of the bags particularly difficult to assemble. We had a number of deviations related to this particular bag, so we're working with the vendor to come up with a better way, and it's an M-fold bag now, so they fold it so it's much easier to assemble for the manufacturing staff. But that process took months. I mean, from identifying the issue, I would say potentially even years, from identifying the issue to going back to them, to giving them feedback, to raising all these complaints, formal deviations to the vendor, to them seeing the issue, working with us and addressing it. And now those particular modified bags are now on the floor, and we're trialing them to see whether they're a big improvement. That remains to be seen. So in terms of anything that we want to change takes a long time. We also have multiple vendors, so not all the vendors provide all of the items for single-use systems. So what I would say we're challenged with is many different connections, literally connections of different vendors' systems that need to work together, and everybody has to work together at the same time to have a successful batch. That being said, we are and have been very successful, but where we do have issues, they tend to be quite impactful. In terms of the concept, and one of the other challenges that we've found has been the more traditional regulators find the whole concept of disposable technology, running multiple products within the same small space, having connectors that go through walls, having totes of material that are not in a GMP graded area, so that it being in controlled non-classified area. A lot of those, they're challenging for regulators. We have had a number of interesting conversations with regulators around how do we control something like leaks, because if you look at a deviation list for our facility compared to a normal stainless facility, we may have 10 times more leaks. So some of those leaks are very unimpactful, but some of them, you know, they can give the impression that we're not in control of our systems. The interactions and how we deal with vendor issues is something that comes up regularly. We have a lot of with much less conversations about cleaning validation, a lot less conversations about sterilization, but we do have a lot of conversations about single-use systems, about leaks, about you know, notifications to vendors and qualifying our suppliers. So that tends to be an issue. Sometimes it's a very interesting conversation. You do your opening presentation, you show your layout diagrams, you talk through everything, and maybe you're 2 or 3 days into an inspection when you actually go up onto the manufacturing floor, and it's amazing the difference that makes. So with some auditors, it's an amazing change. Like they light up and they go, "Oh yes, I get what you mean now, and I understand." With others, they just don't get it and they completely shut down. So we did have some interesting conversations and challenges from regulators around this. The advent of paper-based inspections is not doing us any favors either, because there's a lot of new concepts, and trying to get new concepts to relatively conservative regulators by paper without any visualization of what we're doing has proved challenging. I'm going to stop for a second now and see, does anybody have any questions, because I'm okay. If you have anything, just let me know. Okay.
I think you can drive on. We usually have the questions at the end, but
okay. So in terms of, I'm quite close to the end, Kieran. I'm a fast talker. So just in terms of what the desired outcomes and what we've achieved. So we have built a very flexible facility. We have successfully used single-use system technology, and we have been successfully licensed. So that's really good. We have reduced the workload for MPI introduction. We have had a number of successful PPQ campaigns with no increase in the number of discrepancies or deviations that we'd see compared to a normal process validation campaign. The facilities and the automation, they're validation approved now, and we definitely have a reduction in the number of cross contaminations, and we have a reduced risk for general contaminations due to the closed systems that we're working on. So in terms of what I would say is the next thing I just going to touch on briefly is the leaks and the issues that we have, and I'll talk about these a little bit more. So in a typical month, which was December of 19 is the data I pulled here, we had 5 leaks observed, and those leaks were, these were impactful leaks. They were all resulted in deviations. And there were 5 leaks in this is just purely in the 2,500 litre bag. So at this stage, we're at a very impactful phase of our process. So when we have a leak, the path forward is interesting. So if it's somewhere that's a very small leak, and we've defined what's a minor and a major leak now, you know, if it's not impactful, we don't feel there's any potential for ingress, there's liquid dripping out of it slowly, we tend not to hold off as long as we can without doing anything. If it's a bigger leak than that, we've developed a process now where we can actually transfer it into a new single-use system bag. We have a process where we do that anyway along our process, so it's not an actual change, it's just a transfer. And then we also, what we found is that some of the leaks are quite impactful, and you walk in and the floor is covered in your product, your drug substance, which is very sad and not good from any point of view. The other thing we've seen, so for a 1,000 litre bag, we had a trend of issues which we raised a trend deviation for, and a trend with the vendor as well. And what we discovered was they changed one of their suppliers of materials. Their supply of material had left it with the irradiation of this particular part had caused it to be slightly weakened. And with all of our stock in hand from the same vendor with the same issue, they informed us that there was a higher potential for leaks. And this is where you have these interesting conversations. Because as a QA resource, I'm going, well, what's the risk to the product? And the manufacturing folks are going, what's the risk to the turnover and the turnaround, and what's the financial impact if we have a leak? But what's the financial impact and the impact to patients if we don't manufacture? So these are all the interesting conversations we had in that space. And there was nothing, the vendor had no stock on hand that hadn't used this part. They had to go and manufacture more parts. So for about 4 months, we were operating at risk. But knowing that we had this potential issue, we could put in some mitigations. We had to write an ISPE roads analysis to document how we were getting ahead of the issues and how we plan to deal with them, and to document the risk to the product. So I guess that's probably the biggest challenge that we see. And I suppose what the other thing I would say is we are seeing some of the savings. So just to end on a relatively positive note, what we are seeing savings in is we have reduced Wi-Fi usage, reduced water usage. We use far less steam. Our HVAC bills are way smaller, and we're using less energy generally. Now, in an environmentally conscious world, using an awful lot of plastic and disposing of it, because it's all single use, it's in the name. It's a challenge to make that sound like an environmentally friendly thing, but overall our environmental footprint is actually reduced because our facility is smaller, because our energy use is lower, and we use way less chemicals for cleaning and way less steam for steaming. So it's an interesting challenge for somebody who's an environmentalist to say, well, here's another big huge plastic bag. I use reusable bags every time I go to the shops, but when I'm in work, it's all disposable plastics. So I think that the challenges around that are interesting, but overall it's a positive environmental impact as well as a good impact from a manufacturing point of view. So Just to wrap up, I would say we've been very successful in our mission and we've achieved the vision that they've asked. We have a flexible facility. We have the advantages of being able to dispose of anything that has a contamination or any potential issue. But the disadvantages are we're very reliant, heavily reliant on suppliers, and with single use or single source because of the nature of our business. That can be the challenge, along with the regulatory challenges. So I'm open for any questions or any thoughts that you would like.
Thank you very much for that talk, Miriam. I suppose while the others are having a little think to themselves of what they'll ask, I might just get the ball rolling. Obviously, you mentioned you're very, very reliant on suppliers, in particular for your plastic bags, for your bioreactors and pieces of equipment. Do you guys in Amgen operate a kind of a just-in-time system, or do you have an inventory? Like, for example, if the supply chain was shut down tomorrow, I mean, could you keep going for a year, or would you be in trouble kind of straight away?
So we always operated with 3 months forward coverage of all our, what I would say, high-risk items. We've increased that now because of COVID to 6 months. So that has helped. It's been a challenge, but it's an expensive thing to have 6 months forward coverage of anything. But that's the only way we can operate. As I say, if we don't have our single-use systems, we can't do anything. So we do have a good amount of forward coverage in terms of supply chain. What that does give us, though, if we find a batch that has an issue, we have a lot of inventory, and the vendors are very reluctant, because it's a big financial commitment. They're very reluctant to take them back. So we've had a very good relationship with some of our suppliers. We have a lot of very good suppliers, and we have a very strong relationship with them. We have within ASM, so we all have our network supply points of contact, but even in ASM, for our main single-use system suppliers, we have a point of contact. And that point of contact has a direct line into the companies and the Mercks and the Millipores, and just so we can address issues very quickly and raise the issues very quickly. But at the moment, I think because we were first, we're kind of special to some extent, so we do get treated very well. I think what we need, though, is to make this normal, to make this what everybody does, and then we'll end up with multiple suppliers, a lot of good reliability, and some healthy competition in the market. So that will help.
Yeah, absolutely. Open up to the floor now. Does anyone have any questions this time? I'm sure a lot of you are familiar with that Amgen site in TUAS.
Yeah, Kieran, I'd like to raise something here.
Yeah, work away, Thomas.
Hi, Miriam. Thomas here. You mentioned a few times just to us, and as well, it's the world's first of its class facility. I've been fortunate to follow the development over the many years. I've spoken to some people, senior people from your organization, who not only had been part of the vision, but really lived that vision. So it was really great to follow it along. And even you have that sparkle with you for the single-use technology, and that's also very close to me. My thought is, or my ask to you is, well, now that it's all run up, you have lots of learning. If you would say, well, how would this facility look like if you could envision it 5 years from now, using all the learnings, any kind of perhaps wish list of things you would like to further advance and optimize?
So in terms of the facility itself, what would I think one of the things that we struggle with is training, because when we're doing something, it has to be right for its time. So, you know, when we're training our operators or training our QA staff and getting people on the floor trained up, the cost of not getting it right is very high. So what I think we will do, and we are actually planning to do it, is to build a training facility where we can actually do the mock-ups, where we can actually put in the single-use systems and connect up with the connections and take some of that that risk out of that training process, because it has to be right first time, and if it's not right first time, it's very, very expensive. So I'd say that would be one of the key things. And the other thing is, you know, it's great to be first, and it's terrible to be first. But I think the advantages that we've seen way outweigh the disadvantages. The one thing I'd say is, with our small footprint, I'd make it bigger. So I think if our footprint was a little bit bigger, we'd be able to do more with our floor space. What we find is the systems are single use, but we've a number of columns, we've a number of stainless pieces that we have to use, and you know, we never in our design concept, we didn't consider where we were going to store these. So there's a couple of small challenges there, but generally, I think it was a great thing to do.
Thanks for sharing.
Thank you.
I see there's a question here gone into the group chat. From Wayne Carroll. I'll read it, Wayne, and you can correct me if I'm not getting the point across. But Miriam, you stated that you were able to increase your titer and therefore yield over traditional stainless steel technology. What are the factors that caused the increase in titer?
So it's one of those kind of secret sauce things that was a nice finding. So one of the things is that we have discovered that the cells really like the single-use systems. So they tend to grow better. We tend to get a better growth, and we get better yield by about between 10 and 20% titer improvement. And it's really down to happy cells. I think in terms of all of the yields improvements we've looked at, we can't, you know, in stainless and traditional facilities, and also we're obviously trying to improve yield where we are. The big difference is the fact that the cells really like growing in the single-use systems. So I think maybe stainless doesn't suit them as well. There's a reduction in chemicals, and it's very easy to you know, to modulate and change your process a little bit, just to tweak it if the cells are giving you any messages that they're not happy. So it's quite flexible from that point of view. But I think the real difference is that they like the single-use systems. Which is
yeah, it's the one thing that I noticed about when you spoke about your disposable bioreactors. They're what, 2,000 litre, you said, is that right? Something.
Yeah.
So that seems to be kind of smaller than a lot of the stainless steel, we'll say, bioreactors that I would have seen, which would be more like 6,000, maybe 8,000, 10,000. Do you think because smaller batches, you know, it's more uniform changes in pH, more uniform addition to media, would those be factors?
It could, yeah, absolutely. And I think there's smaller, the idea of having smaller batches. I mean, we've talked about whether we could do it in a bigger bioreactor. I think the risk of a leak would be huge. But yeah, I think it's a good point, Kieran. I think, you know, the fact that your throughput is fairly fast, and we do start the same way as everybody else. We start in a crack a vial and put it into a flask. But once we get to 50 litre scale, we're in disposable bags, and after that, you know, everything is disposable the whole way. Yeah, the control and the size of the batch definitely will have an influence.
Okay.
But we do see better titers as we go along. We see better viability along the way as well. So.
Marab Athin Suresh, sorry for the pronunciation, has put a question in. Could we kindly know some of the major causes of leaks and preventative actions to prevent leaks? If you've got any secret magic wand action leaks.
So in terms of the major cause of leaks is that the bags are just, they can be damaged. They can be damaged in transport. They can be damaged on incoming. They can be damaged in Amgen. They can be damaged in handling. Where they tend to get damaged is along the cracks, so where the folds and the cracks. like any, I suppose any material, you will get distress lines, and we do a deep dive and we do forensic analysis of every leak. I can show you if you'd like to see where we're seeing our leaks, and see if I can share my screen with you. So let's see if I can. I know whether are you able to see my screen?
Yes, I can see that. Yeah.
Okay. So this is a American Millipore bag. It's 2,500 liters, and everywhere you see a red or a green spot is where we've had a leak. So we're looking at that now, and I say you see a lot of them are at the bottom. A lot of those are due to gravity, though, because that's where the leaks will appear. But we do forensically analyze everywhere we have a leak. They do tend to be where the folds are. They do tend to be where we have some connections. We saw a large number of them when I talked about this base panel is where all the instruments go. You get a lot of leaks there. So what causes the leaks, I think it's we don't have a definitive answer, but as you can see, they're all over everywhere. Everywhere you've liquid, you'll have a leak, but they tend to be based at the bottom of the bag, and they tend to be where we will see them is where we've connections or folds.
Okay. Yeah, so it's still a major challenge dealing with leaks. Does anyone, oh, here's another question. Any special storage condition required for these large single-use bags? So from when you first started operating to now, have you put in any special SOPs regarding the storage of these bags or anything of that nature?
So what we have done is they're stored in the warehouse on incoming. They're inspected. Every unit is inspected on incoming. So in terms of how we process them, we've done a little bit of I suppose, a lessons learned. We've learned our lessons the painful way. Anything that's got damaged external packaging, we don't accept. So in terms of how we store them in the warehouse, they're stored. They're not stacked. They're not multi-stacked. We're very, very careful with them. When they come onto the manufacturing floor, they're kitted. They go onto special trolleys, and they're wheeled around. They're, I suppose, a large stainless steel trolley that are specially designed. We have special materials with which we open the external packaging, and they're handled by SMEs all the time. And we have it's just very gentle handling. The inspection process we've improved absolutely over time, but the storage of the bags, we haven't really changed that much. But in terms of how we handle them and how we inspect them, there's a lot of care and attention into that, because the costs of missing something are high. Does that answer your question?
It does, I think. Anyone else have any questions? And also for any of the guys that have put questions into the group chat, if you want Miriam to elaborate on any of those points, please speak up. Sorry, okay, yeah.
Yeah, I don't know. I put a question, but maybe I get cut off. I have a question regarding the new Annex 1. And there is an extended part on the single use where for the first time they talk about the particulate that could be generated in the single use and the fact that they don't wash them before using it in manufacturing, and the vendor assessment that should be linked to that. Is that something that Amgen was doing anyway, or is that something that is driven by Amgen experience, or is some gap, and how do you cover that?
Yeah, so in terms of particulates or leachables and extractables, we have extensive studies to support that. So again, we had to kind of get ahead of that, because regulators who aren't used to seeing single-use systems would tend to be conservative. So we have extensive data on the product contact parts and any potential impact on the product. We've put in, drug substance and drug product release testing to allow us to justify our use of single-use systems all the way along. So we do have a number of filtration steps also to remove anything potential, but we haven't seen any increase in particles from the use of single-use systems. So I know that is a concern.
Thank you.
Okay, another question for you here, Miriam. Could we kindly know what justification Amgen gives to regulators on testing of samples on the manufacturing floor instead of sending them to QC?
So in terms of, this is always an interesting one. Again, it's so testing on the manufacturing floor with the same equipment, it's qualified to the same level. The convenience of having it on the floor is that your samples don't have to travel so far. You get your results faster, and the turnaround time is quicker. We have the justification is that we have quality oversight at every point. We have data audit trail reviews. We have reviews of the system and the process, and it's to the same quality and criteria as we apply to QC. So the location of the equipment is not the issue so much. So the question seems to come up more about oversight and having people who are on the floor maybe having a conflict of interest, and whether it would be in their interest to have a passing result rather than have a result that is unacceptable. But we have defended it successfully. We talk about quality oversight. Quality are independent of manufacturing, and that's always been our role. So it has been a challenge, and some of the agencies are still, I would say, they find it a little bit difficult, but we have successfully defended it. So.
Very good. Another question we have here for you. You're getting an awful lot of, we usually don't have this many questions for Q&A, but for the QA session, but you're getting a lot, so we're really making you work hard today. But are these single-use materials biodegradable? What is the what is the impact on the environment on the use of large quantities of plastic? So I'll repeat that again. Are these single-use materials biodegradable? And what is the impact on the environment on the use of large quantities of plastic?
So they are not biodegradable. They are single-use systems. And what happens to our bags is they are incinerated and they provide some energy for the people of Singapore. So that's how we dispose of our single-use systems. Ideally, you'd like them to be biodegradable, but you know, the risk of having something that is biodegradable is you get more leachables and extractables and potentially particles in your process. So by their very nature, they have to be something that's very unreactive. So I do think there are opportunities there. I'm very green generally, so as I say, there's the pros and cons, or the lower energy costs, the fact that we're not using harsh chemicals to do cleaning, and we have a reduction in HVAC and steam and energy costs generally. So in fact, it is a greener facility despite the fact that we have so much plastic, and it is unfortunately single use. So. Yeah,
well, I'd imagine the less energy that you use for CIP and SIP more than makes up for maybe the plastic trade-off. Just like from my point of view, at the start, I kind of thought that you maybe didn't have to use any CIP or SIP in your facility, and then I think something you said later on, you said that you're using much less steam than other facilities. I mean, do you have some CIP and SIP, or is there no need for any CIP and SIP? Can you just give us a sense of that?
So we have some multi. So some of our filters are in filter housings that need to be autoclaved. We do have some parts that we autoclave, just as a bioburden reduction rather than a requirement for sterilization. But they are we have several autoclave cycles for parts like valves and you know, things that, so we do have a connected process that has is partially stainless. We also have our purification columns are all reusable, so they will be cleaned. So it's the harvest parts are, to some extent, there's some shared equipment in there. They haven't invented a column yet that's disposable, but I'm sure as soon as they do, and then we'll be all over it. But yeah, at the moment there's some shared equipment, but it's minimal. So our cleaning validation is it's very, very low. So we've one very small autoclave and one very small parts washer for quite a big facility. So it's definitely lower than you would see in a normal stainless facility.
Very good, very good. Another question here, and this is question number 10, which I don't think we've had at one of the. I don't think we've had 10 questions on one of these sessions before. But anyway, could you share what is the batch rejection rate in single-use technology when compared with multi-use equipment with respect to contamination and sterility? Any data? So I'll repeat that. Could you mind sharing what is the batch rejection rate in single-use technology when compared with multi-use equipment with respect to contamination and sterility? Any data?
I don't have a direct side-by-side comparison for that. I can tell you we had a couple of contaminations in the 200 batches of our main product and none in the others. So I would say our data set is probably about 260 batches we've manufactured, and we've had maybe 5 that have had contaminations. We found the root cause for those. So and it was a problem with aseptic handling and parts becoming contaminated. In terms of compared to our stainless steel sisters, I actually don't know the data. I'm sorry. I'm not sure of what their numbers are, but it's a low rate. It wouldn't be a major consideration.
Okay, the questions are still coming in faster than I can ask them even. Here's one. What is the turnaround time for a single-use 2,500 litre single-use bioreactor? What is the turnaround time for single-use?
In terms of manufacturer or what?
Yeah, is it, I mean, Bella, perhaps you can clarify that question. Do you mean how long is the fermenting process or how long is the bioreactor?
I'm sorry, this is not the process. Like once harvesting is done, then what's the turnaround for the next single-use bioreactor to start? Yeah, that's my question.
Oh.
Okay, so how long does it take before you can get up and running again?
Oh, a day, not even, like 12 hours. So from one bioreactor to another bioreactor, the turnaround time is very fast. They just get, you take them down and you replace them with another bag and you make a connection. It's very fast. It's like 12 hours would be the longest. So from one, compared to cleaning and steaming, it's way faster. You can probably do it in a number of hours if you wanted to, but like our turnaround times are very fast from one bioreactor to another. And we have a number of trains that go at the same time, so they're not, and you can connect. They're not hard pipes, so you can connect from one to another. So they're easy. It's very flexible in that way. But the changeover time is very fast.
So you're able to go just hell for leather constantly. Here's a question like
That's what it feels like, actually.
Yeah. Here's a question, and I think I probably know the answer to it, but I'll ask you anyway. How do you decontaminate plastic bags after the cell culture is complete? Steam, black steam, or chemical? I mean, you just dispose of the bags, I suppose. Is that right?
Yeah, dispose of them. Unless we have an investigation. If we have an investigation, we use a base to decontaminate them. If we need to send the bag to the vendor for investigation, we need to decontaminate it. We use a base. We flush it through a base, and we can send it then. Otherwise we dispose of it.
Okay, so
And
unless
there's nothing left in it, like it's absolutely like it's just an empty bag. So there's no residual liquid or anything in it. So it's literally just
it's just straight into the incinerator.
straight into the, yeah.
Okay, very good. Okay, well, okay, that seems to be all the questions. Thank you very much, Miriam. I mean, they came very, very fast. If anyone else, I know I was speaking a lot as well, but if anyone else has maybe one more question that's burning on their mind who hasn't asked a question so far, please speak up. This is really a lot of questions. We'll have to set I think you might win the record for the year of most questions asked at Technical Tuesdays. We'll have to make up a little. Okay, if nobody has any more questions, then I think that's it. Listen, that was absolutely fantastic. Thank you very much, Miriam. We can tell by the amount of questions that were asked that I think our audience today, which was about 30 or 31 people, got an awful lot out of this talk. And yeah, thank you very much. And sure, perhaps in a couple of months, you'll join us again.
Thank you so much. I appreciate the opportunity. Oh, I have to put in so there's a lady called Nina who's running for the board in ISPE, so I just have to put in a plug for her. She's an Amgen regulatory person, so I promised her I'd plug her.
So if we have an opportunity to vote for
yes,
Nina to do that, okay.
okay. Please, yeah.
Very, very good, very good.
Thank you. Thank you.
Okay, thank you, Miriam, and thank you to everyone else for joining today. I'm sorry to say, off the top of my head, I don't actually know what next month's Technical Tuesday is. I'm unprepared for this session. Does anyone else know what September's Technical Tuesday is? But anyway, I'm sure it'll be absolutely fantastic. So stick it in your diary for next month. Okay. Thank you, everyone.
Thanks, Kieran. Thanks, Miriam.
Thank you much.
Thank you.
Bye.
Thank you. Bye-bye.
Bye-bye.
Yes.
Bye-bye.